السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. For those that are wondering, in Subhanallah Azim, we're continuing with our final review session for BIOC17, and that inshallah ta'ala will include weeks 6 to 10, inshallah ta'ala. And I know this may be a bit, perhaps I hope beneficial in the sense that we're going through the material at maybe high speed. But the idea that I hope, inshallah ta'ala, with the night ta'ala, if Allah Almighty wills, is that you guys will be able to understand, appreciate the material early on to be able to digest, apply, and understand, especially for novel conditions or situations that you may be. Uh, Posed in the final lab exam. So, inshallah ta'ala, I'll go straight ahead. My goal is to give some amount of time for us to go over the material from the slides, and then about 15 minutes or less, and then inshallah ta'ala, keep the remainder for the lab manual if possible. Bidhnillahi ta'ala. So just give me a moment. Inshallah, we're going to do this shortly. So inshallah ta'ala, we'll begin. Starting now with lab 6. So we went with lab 5. We went with lab 5 going through generation time, and now we're into lab 6 as mentioned before about environmental conditions. And in this regard, even though this may not appear to necessarily be the case, we are dealing with different types of germicides. We're doing further tests of analysis of specific components within specific bacteria. So hopefully, inshallah, you'll appreciate the different realms of which bacteria are able to grow, survive, reproduce, etc., in unique ways with specific features of interest that we'll be exploring. Okay, so as you can see here, we went through actually the chemical germicides, and I hope you know the differences between disinfectants, which normally place on your bench tops or on surfaces, whereas antiseptics normally are used to kill or reduce bacterial growth on living tissues. And that could be on the skin, it could be inside, internally, etc., in different ways. And so you have, the two bacteria that we recall, right, for the differences. And hopefully, if you guys can, try to share the discussion in the comment section, your thoughts as to why we're using Staphylococcus epidermidis versus E. coli here in this exercise for testing different types of chemical germicides. We have bleach, we have hydrogen peroxide, and we have ethanol, and we test them, as you recall, across different concentrations, right? A myriad of them for different germicides, and then for both types of bacteria. And notice, the different traits that these two bacteria have. And so thereafter, right, if you recall, we would have grown them or resuspended them, done what we would have done for a microplate reader and being able to essentially, place the blanks. Actually, it appears to be the blank, yes, across this row here. And then for each group, you would have accordingly labeled your tubes, you would have resuspended the bacteria that would have grown post incubation. And so, what you would have hopefully come to appreciate is a change in turbidity readings, i.e., as we recall, change in absorbance values across changes in concentration of your germicide. And then when it came to antimicrobial susceptibility tests, that's where we used the different discs. As you can see, they had even different labels with letters on the discs themselves and on the vial specific names, be it ampicillin, penicillin, streptomycin, chloramphenicol, or tetracycline. I do recommend knowing the different effects, be it if they're bacteriostatic, i.e. that they inhibit growth, or bactericidal, in that they kill the bacteria themselves or they make the bacteria pass. So that's a separate point. For the beta lactamase test, this is another feature that is important because as you actually can connect the dots from the previous exercise, you can come to appreciate that as different bacteria grow on different antibiotics over time, or with different antibiotics seeded onto your plates, you'll come to appreciate that some bacteria are, essentially resistant, and some are more susceptible to certain antibiotics. And one of the features that actually discriminates or ameliorates their condition, i.e., how whether or not they are able to break down a certain antibiotic or not, is with, for instance, an enzyme known as beta lactamase. Beta lactamase breaks down the beta lactam ring in many penicillin grouped or based antibiotics. So think about penicillin, ampicillin, amoxicillin, usually those psyllin-based, but there are other ones as well. And what this assay depicts, or what this experiment showcases and depicts for you guys or delivers, is that using this disc that aims to be similar into replicating, whether or not you have beta lactamase in your bacteria or not, it's kind of like you having pretty much the antibiotic as this disc, right? Nitrocephalin. Or in fact, cephalosporin, which is another type of like essentially beta-lactam antibiotic or beta-lactam drug. The idea is that it has a ring, and following this reaction of, to simply say, the lysis of your beta-lactam ring through beta-lactamase, you'll actually see a change in color from the nitrocephalin from yellow to red. So this would be an example of a bacteria, if let's say was inoculated onto this disc, it would showcase that it does not have beta lactamase or very minimal, whereas this one clearly indicates, or what it should depict, is that there is beta lactamase. If there was cross-contamination, that's something you want to consider, right? If this hypothetically was your positive control and you would have expected it, maybe you didn't add in the right bacteria, for example. But these are things that you just want to keep in mind. For the catalase test is quite similar in the sense that there are bacteria that may also have catalase. Catalase is an enzyme that can break down hydrogen peroxide and reactive oxygen species to essentially ameliorate the condition in which they're able to survive, i.e. to break down, at least in the case of hydrogen peroxide, which, as you can imagine, is, as you recall, a chemical germicide. It can be broken down into water and oxygen, which is quite readily beneficial for bacteria that are, aerobic, and also having water available indeed as well. So, like you can imagine catalase being very beneficial for certain types of bacteria, and notably that it distinguishes certain bacteria. So some do have the enzyme and some don't. But nonetheless, you could see from rapid bubbling, if it was indeed a positive result, rapid bubbling would be indicative of, essentially catalase presented bacteria, or bacteria that have a lot of catalase, because this reaction where thereby exists where you have, few drops of specific hydrogen peroxide, I believe 2%, or nonetheless the fixed percent that you have, that essentially would increase the oxygen production, which are these bubbles, or essentially the bubbling that you see from your group of bacteria. So bear in mind that you would follow the steps that you recall from the past, right? This would be like a liquid inoculation of your bacteria. Now, hand washing, we did just describe this, but I hope really if you have a look at this video, it's something that's quite interesting indeed. It actually, I recall looking at this, it's actually quite profound in the sense that you can have like a grandeur of many bacteria that can grow over a period of time. And there are different techniques that you would use, right? But just to show you like a bit of the results, at least, like it's quite profound that even, post washing your hands, you can actually see a number of bacteria here, right? Which is quite profound. And then if you, try to, take another step of sanitation, you can essentially see a much greater reduction in bacterial growth. And bear in mind that you're using, like in this case, if I'm not mistaken, blood agar. So you can come to appreciate, even if it's not the same, uh, set of plates, you can come to clearly see that over time, with proper hand washing, be it with, within your knuckles or like on either side, thinking about like high school or, during even COVID perhaps, or, even elementary school when you're learning how to wash your hands with your parents, for example, there's certain ways that you want to be mindful that you're removing any risk of harmful pathogens, right? Pathogens that are essentially bacteria that pose a level of harm to the body or to others, right? Not all bacteria are harmful, indeed, but nonetheless, you could see how a difference in bacterial growth, morphology, shape, and size exists across the different conditions. And so that's something that you want to bear in mind. And then the generation time assignment, we talked about this. Alhamdulillah, Alameen. So this is pretty much completing, lab 6. I briefly just want to highlight though, for those that were wondering, that exercise about generation time, you're aiming to study, the time it takes for your population of bacteria to double. And so there are two ways, either essentially getting the doubling of your cell count, viable counts, direct count, or your absorbance readings from the y-axis, and then just seeing on the x-axis following interpolation the difference between the two x coordinates, right? So if it was, let's say, doubling from, let's say, time point 1 to, let's say, time point 3, then the difference between the two would be essentially 2 minutes. In our case, we know it's 20 minutes, but I just wanted to let you guys know that. I hope it makes sense. If you guys have questions, feel free to also ask them in the respective review summary. And so, yeah, you would have done this for both different types of techniques across different temperatures. And then you would have taken either account this formula or the formula I showed in lab manual or the technique I just mentioned about comparing where the doubling exists on the x-axis. So that's that. And if we go just briefly looking at the lab manual to see if there's any additional material to highlight, you have the use and dilution test, which, by the way, is where we're actually going into the antimicrobial susceptibility test. It is briefly touched upon, and there are conditions that you guys want to be aware of. I also recommend being aware of where you do the centrifugations, the purpose of it, right? Because you want to essentially have a pellet that's concentrated of your bacteria following the resuspension and incubation of your germicide of interest. So just bear in mind that you're following the techniques. A lot of this component for exercise 6 or week 6 does maintain specific conditions for methodology. So just bear those details in mind for your lab exam. And similarly, the expectations of your blank, your absorbance readings, the initial time points, usually between 0.3 to 0.05. And then just like what I talked about, like the certain dilutions or use dilution test and the pretty much the conditions that are standardized here, even though we couldn't adhere to them, these are things that you do want to note because they also impact the level of inhibition, i.e. how large your zone is and the morphology of your zone, which is something that you want to keep in mind, right? Because they actually influence the different conditions you see here, i.e. if the organism indeed is susceptible, intermediate, or in full resistance, or essentially resistant to your antibiotic. And there are the different conditions mentioned here, the plates as well, right? Having a pH that's 7.2 to 7.4, which is ideal for mesophilic bacteria. And so you just want to bear in mind the different conditions that are mentioned here as well, and the density in which from the starting tube that specific bacteria will be able to grow in and thus be able to readily be observed post-incubation, the impacts that those antibiotics of interest have on your bacteria. Also bearing in mind the depth in which those antibiotics are from like essentially the discs themselves are able to diffuse through the medium as the bacteria are able to grow therein. So that's pretty much, pretty much what I want to highlight. And this is something you want to bear in mind for cephalosporins, penicillin-based, antibiotics. There's a beta-lactam ring that gets broken down, right? Yeah, pretty much this is it from the additional material. I want us to go to now, maybe if there's anything else, I'll just have a look. Effects of hand washing, right? And you want to bear in mind the different details that we've highlighted. Perhaps even if they were virtual, you still want to know the different, conditions that perhaps it could be replicable to what you would do in terms of, methodological analysis for your lab exam. So think about the different components. If you see additional questions, even though I may skim, like may pass by them, consider the different questions that may come up in terms of asking yourself, like here about the different components and impacts that certain, steps in your procedure may have on your results down the road. So if there are any questions, feel free to pause, feel free to let me know. Um, inshallah, let's continue with lab 7. So lab 7 goes into the lethal effect of UV, right? Our FLR, if you recall, right? So we had different sets of bacteria. We had E. coli, we had Staphylococcus epidermidis, we had B. subtilis, or Bacillus subtilis at 24 hours and 7 days. Hopefully, we know the difference, right? E. coli and Staphylococcus epidermidis, they differentiate on the gram stain, whereas B. subtilis at 24 and 7 days, hypothetically, would be able to be discriminated using the endospore stain, which is interesting because those two stains were stains that you used for an unknown assignment. So hopefully a lot of these different components integrate well with what you're able to understand, inshallah ta'ala. And with that, you would have essentially common spread your bacteria, 1/3 turns, inoculations, right? Um, and then you would have placed them under UV, right? I know there were a lot of sets of plates that we were dealing with, but hopefully, even just for this exercise, inshallah, you would be able to understand the limitations, the conditions, and the different expectations that you would have had across the different bacteria based on the traits that we've described, right? And the features that they have potentially against against UV exposure or UV. And so in terms of, the impacts that UV has, right? If we have a look at this figure, right, you have the pyrimidine dimers, right? Bear in mind, you know what the pyrimidines are. That's something to just, make note of. But essentially, those specific bases are essentially dimerizing within the same strand, like shown here, meaning that obviously it would impact, not just bacterial replication, but even bacterial transcription, gene transcription, right? So think about the impacts that thus UV would have, especially when you're trying to, let's say, grow bacteria, right? Because they need also cell wall synthesis, they need protein synthesis. And so if specific or multiple gene sets are being interrupted or, dimerizing irregularly or essentially dimerizing, you have the risk of essentially not just mutations and reduction in expression, but even cell lysis potentially or like unexpected consequences, perhaps those that could be indeed lethal. So consider those details. And when it comes to the platelets, I know this was a bit contentious, but nonetheless, in terms of the lids, at least, you wanted to make sure that we followed a aseptic technique. It would be artificial, but I hope actually let's go. Like if you look, sorry. If you had because you would be actually opening up your plates of interest, it would be detrimental to have essentially the lid opened up with exposure either on the bench top or to the airborne environment, right? Which is why I was also concerned about it in this regard, right in front of you. But nonetheless, that's one of the rationales. And then if we move to the second page, or like the second exercise or third slide, you can appreciate then the impacts of repair mechanisms, right? Or UV radiation versus repair. And so you had Micrococcus roseus, you recall, it's a red looking bacteria, right, across the slant. And so you would have added in, saline, rub it up around your bacteria with the cotton swab, and then smeared accordingly with your swab across your plate with 1/3 inoculations each time. So this wasn't really like, by the way, I mean, it was more like a manual form of spread plating because you weren't using the L rod, but you weren't necessarily streak plating across like different quadrants, but you're doing all around, right? So this is like a manual form of spread plating, but you would have been inoculating with a sterile swab. Just bear that in mind as a difference. And thereafter, you would have used likely a star-shaped mask, right? And the idea is that that would have controlled, and actually, even though for exercise one it wasn't highlighted, you had, as you recall from your FLR, a half side mask, which was side B, for example, and then an unmasked UV, like exposed essentially side for bacterial exposure to UV. So nonetheless, you have that mechanism, and something that, or something to bear in mind in terms of the setup is that you would have, unlike in the previous exercise, done them for like 0 to 5 minute increments all the way to 30 minutes. Sorry, 0 to 30 minutes with 5 minute increments roughly. You would have had as well, you know, in this regard, shorter incubations, right? So that makes sense if you wanted to see the impact of repair, because obviously you're growing these bacteria over a longer duration post incubation overnight. And so, inshallah, even though like the UV exposure is like 30 seconds to 3 minutes, you could come to appreciate the different impacts that you have post incubation, and even during the incubation or exposure, the effects of or the adaptability or the potential for repair for these different sets of bacteria or the same bacteria across different conditions. And so you had the lid off, direct exposure, lid on, was essentially preventative, almost like a control without direct exposure. Even though they're differed in across time, one extension could even be having the 30 seconds lid on treatment, though technically you wouldn't expect too much of an impact. Nonetheless, you had the different treatments, the 0 minutes indicative of no UV. But then what's important and how that connects to plate 6 and 7 is the differences in incubation, being at light or at day, light or dark. And you know that there are differences, right, between light and dark repairs. So do note to those differences when it's applicable to your exam. And then for the applying the mask I mentioned earlier, but essentially, even though you may have these sets of controls within your single plate, it's also beneficial to see if that outside, right, in the essentially shaded or masked regions, if it does indeed serve as a control, i.e. you still find growth, whereas in with the UV exposure, there's limited growth, or if there appears to be a few single colonies that could be resistant or could have repaired following UV exposure post incubation or during incubation. Um, so incubation like post exposure. So you have nonetheless a slide that's similar to Micrococcus roseus, having a reddish hint, but this is more, more red indeed. And so something that's quite distinguishable actually, and maybe some of you would have seen this or appreciated that, there could be the appearance of white colonies. So even though you may not be like, like I want you to actually think about these details because as you can see, the phenotype has changed for these colonies that likely are resistant, meaning that if they're resistant, it could also indicate that perhaps they actually like with the UV exposure, and you could see at mass, even though this is in a range of like seconds, 30 to 60 seconds, there's indeed a drop in colonial growth from viable cell counts. But what you can see and appreciate is that there are a distinguishable number of white colonies that depict resistant, UV-resistant Serratia marcensis, or marcens. So you want to bear in mind that the colonial growth resistant bacteria does increase or exists, and thus could depict that even despite the pyrimidine dimerization, they were able to repair, and with with photolysase like repair, and the reason I bring this up is thus you can appreciate that perhaps um, these bacteria will be able to grow post UV exposure. And then you would have seen the results of your last week's material, right? Which you just talked about for week 6, chemical germicides. You would have done your presentation as well as, bearing in mind the zones of inhibition from different antibiotics for both plates that you analyzed, so E. coli and Staphylococcus epidermidis. And so I think that concludes. Yeah, if you had questions, generation time, you can mention them as well. So let me just briefly go over. I don't know if we have time. Briefly, I just want to highlight, the reflective practice here. You have 4 different questions. Some of them maybe you can actually imagine. I've talked about like question 3 briefly now, and maybe question 4 you can understand from what I've talked about from the previous seminar. In fact, for questions 1 and 2, and then question 4 could be incorporated with week 6 and 7. But I hope, inshallah ta'ala, you guys have an understanding of this. And if you are, let's say, missing, specific details, or maybe I went through things a bit too quickly, you can also make mention of them in the captions, or even before I mention that, if there are, like, if you haven't been able to, like if you have questions or if you have answers, responses to this, even though I can't answer them as of yet, you can share them in the comments shown in the YouTube video with the night time. So yes, and think about the connections, by the way. See, you have conjugation being mentioned and referenced in terms of the strains that are being used, right? Why is it necessary to use an oxytrophic strain when introducing new DNA into existing bacterial cells? Think about the application, right? Like, why would you? So think about, and you look back into the manual to see this further. But remember what I mentioned before, right? The differences in specific, or like look back into the points that are mentioned in the manual, points mentioned that I've mentioned in our weeks together, and even from the slides from our seminars today and from before, or from the previous review, didn't I, Tala? So with that, didn't I, Tala, let me just briefly go through the manual. I do want to highlight, UV repair mechanisms we've touched upon. So let's go now into actually highlighting. You don't want to know that there are different types of UV. Actually, I want you to understand, the different mechanisms that exist for the pyrimidine molecules, right? So this is cytosine or thymine, right? Uridine is technically a pyrimidine as well. But essentially, you have C. Normally, like if you remember complementary base pairing, goes to T and C goes to G. But what happens in this case is that you have hydrogen bonding between the cytosines and the thymidines, or together, cytosine and thymine together, inversed either way, and that also impacts essentially DNA and bacterial DNA replication or transcription as well, and that can actually have catastrophic effects on your bacteria. We talked about the differences in time points for UV exposure, and then radiation damage and repair is something that I wanted you guys to look a bit more into, because this is something that is limited in discussion from our labs together, but you'll hopefully integrate the material from what you've learned from lecture. So you have from the light repair, for example, of thymine dimers, use photolysase. Photolysase is essentially going to snip, first off, actually, if you can see here, I think you guys can see, but essentially think about it like this. This is an enzyme, right? Photolyase. So it's going to be activated or active upon light incubation. And so as you can see here, it becomes active following UV irradiation, and that's where the thymine dimers exist, right? And you can actually see how the nascent, like the adjacent or anti-parallel nucleotides are left essentially without hydrogen bonding. So you can just imagine thus that the stability of the DNA itself is jeopardized or in question, and so or impacted. And so what you can see is that as you go down, um, essentially you have the monomerization. So you have like the splitting, the lyase or the lysis of a hydrogen bond between the dimers, and that splits them up. And then essentially you can use other enzymes to like ligase to essentially return the hydrogen bonding between nucleotides. And so that's where, even though it may, cleave the cross-link of the dimers to monomerize, you still use other enzymes accordingly to continue this this repair. And then when you have, and actually I think it's also highlighted here. Let's see. And this is given as a case scenario. A helicase acts to remove the nucleotides in a damaged region. Yeah, and that's something I wanted to also highlight. If that's another repair mechanism, the nuclease cleavage or excision repair mechanism, where essentially you actually have the snipping, right, which is used with this endonuclease, UVRABC, right? So bear in mind the acronym shown here. The idea is that it would have removed a portion of the DNA strand that had the dimerization of nucleotides, and thereafter, essentially, you would use DNA polymerase to essentially return. Think about like DNA replication. Essentially, the primer considered either here or this end, depending on the direction, 5' to 3', if you recall. And so, inshallah, the idea is that you would have had DNA polymerase continuing to polymerize pretty much free nucleotides to continue to make a sealable full strand that would essentially complement the base pairs shown on the antisense strand or on the other strand. So bear that in mind. That's actually interestingly a good note in contrast to essentially the light repair mechanism, because light repair deals with DNA photolysis, and then dark repair uses the excision repair of the endonuclease. So bear those details in mind, see those impacts based on the limited factors that exist, right? Light versus dark. You have the presence of dimers in both cases, but how they're being lysed or how they're being, how the strand itself is being impacted can differ accordingly for either repair mechanism. And light, the strand remains intact, but the dimerization of the two nucleotides or pyrimidines is ceased or lysed, whereas for the dark repair, you actually have a strand that's physically removed, the one that has the dimers, and thereafter replaced with following polymerization. So I hope that makes sense. And bear in mind that UV safety, don't look at the lights directly. And try to answer these questions. I want you guys to answer at least 3 questions for each seminar. Post them into the comments if you haven't done so, inshallah ta'ala, or if you haven't like responded or communicated such via the online synchronous seminar last week. So inshallah ta'ala, I hope, or even previously, that you'll be able to answer these questions and consider them integrating the concepts that we've described thus far. Any questions? Or at this time, inshallah ta'ala, we can make our way to week 8. And so week 8 material, environmental factors, I talked about this, but I want to highlight that you have actually 3 different major constituents or constraints or factors, really, variables of interest that you're trying to control or manipulate based on the bacteria that you're analyzing, right? And in our case, multiple bacteria per group in the case of temperature. So think about it like this. Bacteria are able to grow, right? We learned about generation time, proliferation, repair mechanisms. We learned about differential staining techniques, spread plating, streak plating, viable counts, plaque counts, etc. So think about those components and think about how the life of the bacteria. The bacteria, as we talked about for generation time, they sometimes have to attenuate to their novel conditions or their new environment. So imagine if that environment has a higher temperature or a lower pH or a higher salinity, those in turn too also impact the growth of those bacteria, and sometimes they have to regulate their growth versus synthesis versus repair and homeostasis. So there's a lot of things that are involved, even though we haven't really touched upon them integratively. But I hope now you can, based on what I've talked about and what I've tried to appreciate and hone in for you guys to elucidate throughout our practicals together. Right. So let's go forward, right? So you have, your inoculation of, a one full 10 microliter end of your loop, and then you would essentially following resuspension. Remember resuspension, right? Think about limitations as I described them to you guys, right? Because there's certain things that you guys can already know based on what we've talked about, based on what you've recorded, based on what you can elucidate therefore, in prep for your lab exam. For example, Bacillus stereothermophilus, right, is a bacillus. It is, it's a gram-positive bacteria. And the thermophilus gives you the hint that it's thermoliking, right, i.e. it likes the thermal range of higher temps versus the cryophil, Micrococcus cryophilus, is more on the colder end. So it's a saccharophile. So bear those details in mind. Also consider how, these B bacteria are essentially from the same genus. So you can appreciate that even within the same genus, that there are different species of bacteria that exist with different thermal optimal conditions. Same for other environmental conditions. Some have catalase, some don't, right? As we talked about for the Streptococcus family, right? As you saw, some Streptococci do and some don't, right? And so I want you guys to think about these different properties of bacteria and how they optimally grow in different conditions. And so as you can see here, we had a myriad of them from 10 to 60 degrees. And so what we're able to do is essentially following the inoculation, we would actually either we'll incubate them and then see the changes overnight or technically the following week with qualitative observations. And then technically thereafter, what you could do is, and what was done in the past is actually using the SPECT 20 to thereby essentially see the turbidity readings, post, overnight or a week incubation. Something to bear in mind is that we also did this in reps, right? So you did want to see homogeneity, or i.e. like the same result ideally in both reps. Generally speaking, there may be a small gradation, right, of the difference. But generally speaking, right, like if you, followed the right protocol and you have considered, avoiding limitations, then inshallah ta'ala you have very sound results. And based on these results, you can imagine like at room temperature, maybe the E. coli will control doesn't have any bacteria. So between the four sets of bacterial conditions, maybe E. coli goes well, right? 37 degrees, 20 minutes generation time. So it likely was in log phase, producing quickly, especially after, inoculating it for over 24 hours in this tube. And you can just imagine the impacts there after for like the thermophile, right? Because it likes the hotter, temps. So maybe it has a reduced, essentially turbidity or reproductive rate, wherein the generation time would be longer, take longer to essentially follow through binary fission. And then the micrococcus similarly. Maybe the E. coli and B. subtilis were roughly the same. But then when you go into higher temps, probably the B. stereothermophilus would be more would have a higher would essentially grow faster, right? lower, in contrast, the Micrococcus cryophilus will grow less, right? So it have a higher turbidity here, whereas a lower turbidity in the other ones, especially in Micrococcus microphilus. And then the converse for the opposite readings, if you went to colder temps, then you would imagine that there would be a higher turbidity here, and then the other ones would have it more lower, and especially lower for the Thermophilus. So think about those constraints or the different ranges and the optimal properties or optimal conditions in which those bacteria could grow, which reflects their generation time, right? And that also reflects their turbidity readings that you would expect over time. So think about those different conditions and assume that you're looking at them at a specific for a finite amount of time, because technically over, let's say, 48 hours, all of them maybe like reached a very high turbidity, and absorbance readings may not necessarily discriminate them quite readily, which is something you want to consider also based on your results from our former week 8. So bear those details in mind. Then you have pH, right? This is interesting. It also connects us to, the Snyder test once again, because you had low pHs to start off with for optimal conditions of lactobacilli. So nonetheless, you have a range of pHs, right, that you could explore. Even though you may have done them differently for your practical, I just want you to see it based on the ranges that we have for different bacteria, and then essentially doing this across the different bacteria that you see here. So imagine like, you have 3 sets of bacteria plus a control ideally, and a control could be just for each pH, no bacteria included, right? So just imagine like that. You have essentially all of this, like all of the ranges, and consider the growth and as I mentioned, the lactobacilli, right? Think about the different bacteria that you would have likely used and connect the dots with their applications and implications that you expect from the results, and perhaps limit, like, understanding thus how you can best reason and predict the results accordingly, as from what you expect. So, or let's say the lactobacilli. Actually, if we go to Staphylococcus epidermidis, right, we know that it's likely in a more mesophilic range, right, probably between pH of 6 to 8. So expect that the turbidity would be higher here, right? Higher absorbance readings, lower transmittance, higher turbidity, like a quicker binary fission, which is like a shorter generation time, or simply said, higher turbidity, greater absorbance readings, right? Or more essentially more growth, right? And then for lactobacilli, probably around this range, right? Because you have the Snyder test, pH is going down from 4.4 to less than that, in fact. So expected to go lower from 4.2 even, and then thus you can expect that these lactobacilli could grow, and that's plural for lactobacillus, but essentially expect these rod-shaped bacteria to grow and have higher turbidities at this range, whereas lower turbidities as you go further up in level. And then in contrast to the Saccharomyces cerevisiae, so I want you to think about this one and think about fermentation, right? Think about fermentation for a moment, because what are you doing? What is happening when bacteria metabolize, right? Let's say it's yeast and fermenting ethanol. Ethanol, is it acid? Is it a base? Think about like what you would expect therein, right? And so would it likely then be at a higher pH level? And then thus, what do you expect then in terms of growth, generation time, turbidity, and then consequentially the essentially change in growth across pHs. So think about this. This could be one of the questions that you want to respond to, the impacts that be for temp, pH, or the following slide, the changes that they have in turn for the growth of your bacteria. Now let's go to osmotic pressure. So that's the third one. And you can see here, and there are different components. And I think this is good to actually highlight here like to describe a bit more. So like let's have a look here. you have normal cells, right? Like this doesn't even have to be a bacterial cell. It could be a circular RBC, red blood cell. And so if you're in an isotonic solution, right, i.e. the amount of solutes inside and actually I would say not even just the amount, it's actually not the amount, it's the concentration. Concentration, if the concentration of the solutes outside is equal to the concentration of solutes inside, i.e. because the volume here is smaller, you expect a smaller amount of solutes, right? Not the same amount per se. So if it's isotonic, i.e. both concentrations extracellularly and intracellularly are the same, then you expect essentially that the plasma membrane to remain integrated and like to essentially have a stern integrity, like to have good integrity, and to be sturdy, or to be sorry, not the well, yes, the plasma membrane to essentially remain um, like um, integratable, like to have good integrity. I'm just trying to think of another word for it, but to remain strong. And then in contrast, when you have hypertonic concentrations, let's say of NaCl, right, that's outside your solution, which is often the case across these different conditions. Then the idea is that with, water flow, like you think of osmosis, but we could just say as like water flow outside, right, to match up with the increased demand for water to essentially equilibrate the different um, concentrations of solutes inside and outside. Then the idea is that the plasma membrane is actually going to shrink. And think about it for a moment. So you have a lot of solutes outside, right? It's a higher concentration of NaCl outside than inside to start off with, right? And because if you think about it, bacteria, they're not going to have excessive amounts of solutes inside, especially if they form waste products. So they're going to be outside mainly. And then some, indeed, nutrients, metabolites, etc., electrolytes inside. But with the abundance of NaCl, right, outside or presentable, you have water that's with osmosis, water is going to come out, not NaCl coming in, right? It could be toxic, as you can imagine, right? To have solutes just bursting through the cell and like being maintained within a cell while maintaining other metabolic pathways. So the idea is that water is going to go through, and it's more fluid after all. And so think about there too that water is going to be, coming out of your bacteria. So if it's coming out and the cell wall is normally fixed, is what I was trying to refer to, then pretty much the gap between what solutes, or sorry, the solvent, i.e., like your cytoplasm or your nucleoplasm within the cell, because that's abundant with water, it's actually going to shrink, right? So that's how you actually have the plasma membrane in turn also shrinking, or at least shriveling, because the plasma, the whatever's inside, i.e., the solvent, is actually coming outside to make up or compensate the increased solute concentration within your solution. So think about that. I hope that makes sense. And in fact, that could definitely impact cell growth because now the plasma membrane isn't able to neatly essentially fizz or separate when um, needed to replicate. And even you can imagine the reduction in size for other metabolic processes that are based in the cytoplasm. So there are a lot of impacts therein that could exist following plasmolysis. So that's how detrimental, solutes could be perhaps in osmotic pressure, which is also like if you think about it, like if some of you may season, let's say steaks or like, certain types of foods or beverages, like with lime, for example, you can imagine the impact that thus that you can have on bacterial growth or the impact that it has on bacterial growth indeed. And that's similar to the case of, our germicides. So connecting back 2 weeks before, the impact that thus you have like osmotic pressure, be it of ethanol, of, bleach, because bleach is also a hypertonic solution, hypotonic solution. That these include solutes, right? So think about thus the implications of the ions separating in the solvent, and thereby the impacts they have on the survival and growth or reproduction of your bacteria, post-binary fission. And so think about that. If you wanted to have a control, it could be at 0%. It could also be at all of these. So you can have a control technically of no concentration of NaCl or of your solutes of interest, which reflects essentially a homeosmotic pressure because you would assume that it would be isotonic, right? So osmotic pressure is essentially like the pressure that the solutes have on living organ on your species, on your specimen, for example. It could be living or it could be non-living as well, but the idea is that they could have impact. So osmolarity, osmotic pressure, these are like, in this regard, concepts that were integrated um, interchangeably. And so, and or salinity as well. But salinity is usually referred to like NaCl, like saline as an example. But nonetheless, you have a control that could exist at 0% here across the three different bacteria, or even having for each of those tubes not a bacteria. So like those three bacteria, right? Technically across the conditions with your control at 0%, plus a second, like a fourth tube that has no bacteria added in, right? So technically, you have two sets of potential controls. One that serves as a positive control, right, at 0% because bacteria could thereby grow, whereas another serves as a negative control wherein there is no bacteria added in, so you expect no growth, and that would reflect a zero reading for qualitative observations similar to across the other conditions, and then quantitatively you would expect that your absorbance reading should be similar to the blank, right? Because technically both of them would serve like similar uses with very similar absorbance or equal absorbance readings because they typically would be the same media solution. So that's something you want to bear in mind. And by the way, you would also blank your tubes for SPEC 20 analysis from the very beginning, as described previously. So now let's go to thermal death time. Actually, let's pause for a moment. If you want to go back, that's okay as well. Um, I think maybe we can have a look briefly at the manual, because I think this is something that wow, subhanAllah, thermal death time just came in. And so, like what I talked about before, even though I mentioned, mesophiles in reference to changes in, and maybe just to clarify for you guys, the different classifications. Um, sorry. You have the different classifications for bacteria. So you have the different classifications of bacteria across the different thermal conditions, right? I mentioned saprophiles, lower temps, like cold temperatures, right? 10 degrees, 0 degrees, -10, -20, 15 degrees below this usually. And then you have mesophiles that are at the more optimal ranges, 20 degrees to about less than 40 degrees, like 37 degrees are mesophiles, and then thermophiles are usually over 40, 45 degrees and beyond, to like 80 degrees. And you have like, and interestingly, the impacts that those have, not just on the cells, but even on the growth mechanisms of those cells and the organellular or subcellular properties that they have. That's a beautiful point that you want to keep in mind, like biochem wise, or and just understand the material. But you have then pH conditions. Classifications are noticed here. Low pHs where bacteria are able to grow. Those bacteria are considered or classified as acidophiles, or pHs that are near a pH of 7, and bacteria that grow optimally here. those are considered neurophiles. And by the way, when we're referring to these different classifications, we're assuming that's where they most optimally grow for specific bacterial species, not of the genus, but of species in particular. Because we saw, Bacillus stereothermophilus versus B. subtilis, right? So there are differences, but you want to bear in mind that, that as they have maybe differences in thermal conditions, the different classifications can be specified and found here for the different optimal growth conditions. And you have neutrophils, once again, they have, just to clarify, pHs that are, near a pH of 7. And then you have the alkalophiles that grow optimally at pHs higher than 7, around like 8, 10, and more. And then you have like for osmolarity or salinity or osmotic pressure, you have bacteria that are considered either osmotolerant, i.e., that they're able to survive and grow in different like extreme ranges, or i.e., they can grow at high ranges of osmotic pressure, or as with solute increases in solute, sorry, higher osmotic concentrations or solute concentrations, or we'll say osmotic pressures. And then you have the or osmotic conditions. And then non-osmotic osmotolerant bacteria are essentially bacteria that grow at lower osmotic concentrations or conditions. And that would essentially indicate that they're more sensitive to increases in osmolarity generally. But nonetheless, you normally assume that if you're not osmotolerant, they grow best with less osmotic pressure um, from the environment or with the environment. And so you have that. I think it's better now we actually go to thermal death time, talking about it a bit more. And you can see the implications, by the way, of like different unique areas, different gradations that are also highlighted near the end of today's practical, if not the following practical. But you can also bear in mind, right, you have already hints from the lab manual, different types of conditions that bacteria are able to grow in via of temperatures, pHs, of salinities. So these are things that you want to keep in mind, and then different growth curves or the rates in which that they're able to grow, which also reflects neatly generation time, right? So if let's say you have an acidophile that's growing here at a pH of let's say 3.7, then you know that your generation time at that time would be, or at that pH would likely be lower, lower in the sense that it's like it's shorter. So just to clarify, when I'm saying like a generation time that's shorter, i.e., instead of being, let's say, 20 minutes, hypothetically, it could be 15 minutes. That means that it's rapidly dividing even more further, right? So that means it's going faster, reflective of this growth rate. That's optimal at its highest or peak here. And then if you go down in either way for all in different classifications of the bacteria across pH or even other temps, then you'll notice that they go down in unique ways, typically in which the growth rate decreases and thereby generation time takes longer to essentially replicate over time or double over time. So think about the different components. Think about what I've touched upon. I hope you guys will be able to understand and appreciate those differences and the environmental conditions. And you can answer, any follow-up questions that perhaps you haven't sent to me as additional comments in the YouTube video or in the following once we conclude with the night time. Yes, and you also have extremities. So you have like extreme halophiles. Halophiles that essentially grow up at very high, right? NaCl or salinities, and essentially, they're still able to grow, which is quite unique, and they're very osmotolerant indeed. So these are things that you want to keep in mind, the different, nomenclature terminologies that are, quite important to elucidate and to compare because you can be told about a halophile, which essentially is able to grow and like modest saline conditions, or osmotic tolerance as a different set of terms that reflects a similar phenomenon. So just bear in mind that you know the differences, the different conditions the different levels in which they optimally grow. And that can hopefully give you an idea with the examples that you know of E. coli, Staphylococcus epidermidis, S. cerevisiae, etc., how they would be able to grow. And inshallah ta'ala, we can move and inshallah ta'ala touch upon exercise 4. So this exercise, let's go to the slides back now here, where, like think about the generations on assignment, right? Each group had its own flask, right? I believe also like 45 mils of sterile nutrient broth, and then you added in 5 mils of E. coli. So that would have been, by the way, a 1 to 10 dilution, just so you're aware. Whereas this is like a 1 to 50 dilution. Nonetheless, right? And obviously, you can imagine the differences and applications of that because, at that time, a 1 to 10 dilution would be better for us to see the growth logarithmically of E. coli, whereas here we're actually trying to see the opposite, right? So this is kind of like the opposite of generation time because the population growth curve, being if you were to do viable counts or of the absorbance values for thermal death time, is going down, right? You're increasing the exposure to extremities, right? So think about it like that. It does connect to UV exposure a bit. And indeed, you can actually apply this in a similar way, right? If you had technically a set of flasks like the ones shown here, and you expose them to UV, and then you did viable counts, like plating them thereafter, following UV exposure. This would serve as a form of direct counting, right? Or direct method for counting bacteria. So think about, by the way, folks, even though it may sound like, practical, you'll see that I hope actually right now you've come to appreciate the integration of, weeks 5 to 6 all the way to 8, right? And we've actually connected the dots between weeks 1 and 4 to even week 6. But I hope that you guys are able to appreciate the different conditions and different, integrations of different lab concepts because a lot of this is applicable not just to your exam, but to your better understanding and appreciation of within and beyond the sciences, right? I believe like, all of this is indeed, part of just a small portion of the creation. And there's indeed a higher power that is fully wise and fully attributable of all glory, that indeed knows best in every dimension. So in every discipline as well. So think about, the integration, the appreciation of these different components, concepts, and, ideas and theories that could perhaps give you better or if valid, can give you greater appreciation for the deen of Islam. So, inshallah ta'ala, I hope that's something that you will appreciate. And once again, see, we talked about this, I believe, in week 4, serial dilutions, week 4 or week 3, that, nonetheless, like you have a serial dilution, we're going through the methodological integration here again, right? We're going back into the past, doing serial dilutions, right? Across different tubes, right? And this was with your thermal death time. You would have done this at 0 or 5 minutes at 50 or 65 degrees. Bear in mind the bacteria that you're using once again. Just like how we're specific in details for our figure captions, I want you to bear in mind those same details when you're prepping your lab exam, be it if there's any figures, diagrams, or tables, etc., or even when it comes to your understanding of questions, when it comes to the methods, the species, the different experimental conditions, treatments, expectations, predictions, observations, limitations, extensions, implications, applications. There's so many things that indeed are falling through integrations with the I5 approach. So I hope, Bidinai Ta'ala, you stay true to this. And if you have questions, have a look at my previous seminars as well, from review and from our golden age as well, Bidinai Ta'ala, following our golden lab. So inshallah ta'ala, let's go forward. And so you can see, right, 1 mil inoculations or 1 mil aliquots to 9 mil saline blanks. Bear in mind even the concentration. I mean, it hasn't been mentioned, but these saline blanks are essentially still allowable or optimal for growth, or like the growth of bacteria can still exist in this regard. Nonetheless, you're primarily seeing, at a finite amount of time, you're not and this is something that's an assumption, but you could technically, account for, the time that, from where you essentially do these serial dilutions to the time that you place them onto the plates, there could be the potential of, bacteria Um, Actually, to highlight into the serial dilutions, you would have just done these 1 mil inoculations repetitively, serial dilutions, and then you would have noticed that the 1 mil dilution in a 9 mil saline blank leads to a 10 to the minus 1 dilution in that tube, and then that repeats for 1 mil into the of a 1 out of 10 to the minus 1 dilution into a tube with already 9 mils of saline blank. That would have essentially, or a 9 mils of saline solution, you would have essentially done the 10, gotten a tube to 10 to the minus 2, 10 to the, and then you would repeat that 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. And then importantly, you would have taken 0.1 mils, which is attributed to that 1 mil, right? So it's like 0.1 divided by 1, that's 10 times less, or 0.1 essentially. And so you do essentially 10 to the minus 1 times 10 to the minus 1, or 10 to the minus 1 times 0.1. And you do that across the board, across the different tubes. So you would have expected a one lower or a logarithmically lower, consider logarithms an application in this regard for the final dilution factors, which by the way is appropriate to note. Like if let's say your colonial growth, right? Some of them it makes sense that if you have colonies that are too many to count, let's say here, it makes sense that, let's say this is like 3,780, right? This would technically be too many to count. Probably see too many colonies following incubation, right? But then slowly you'll see, oh, like let's say it's 378, right? It's like 10 times less. Still a bit too many to count. But then thereafter, you'll get probably 38 because divided by 10, you wouldn't just say 37.8 colonies, but maybe unless you were averaging, but you would say, let's say 38 colonies. The idea that I'm saying this is so that you can appreciate that the 10 times reduction in dilution should impact the 10 times, hypothetically, the population that you're able to count of viable colonies. So the viable cell count thereby should, you can predict, go down by 10 or tenfold. Nonetheless, but you have like these different sets of tubes. You would also have a blank for SPEC 20 analysis. But we normally did this with a microplate reader, right? Nonetheless, you would have this, and bear in mind you're doing this at 50 and 65 degrees, so this is a difference, something to bear in mind. And then thereafter, you would have essentially actually another point, two separate points, but one is that you would have also done this directly with 0.1 mils added into the 10 to the minus 1 plate here that's shown on the left-hand side. Benefit of this is also to ensure that you have a stock, kind of like a stock plate, even though it's 10 to the minus 1, you can appreciate if there's, let's say in this semi-control, if you find no bacterial growth, then it's possible that no bacteria was inoculated into the stock itself, like i.e. there's no bacteria here, or when you actually did the pipette, you didn't add in bacteria. Second option, that's the second point, right? When you did the transfer. The third point is that it's possible that there was contamination, right? And so it's possible that perhaps the bacteria were lysed, right? Could be with say about lambda phage, right? There's many, like, even though we're not dealing with the lambda phage here, but it's possible that there could be cross contamination. There could have been nonetheless, those two other options exist quite readily. So what I'm just trying to say here is that you have the dilution, the serial dilution shown here, and then the implications of the plates with 10 times less of a dilution factor. And you have the second point of, doing this across time, right? So I talked about this, right? You're doing this even for absorbances for the microplate reader, but you can appreciate that as soon as I added in, by the way, the bacteria, right, that would have likely be, assumed time point 0, and then we would have waited ideally 5 minutes. But I do want to, if I go now to, and that would have been for viable counts, but if we look at the microplate reader, and this is something I just want you guys to pay close attention to, understand the setup. For those that were wondering from before, that you have essentially column 1 as just your blank, right? And then and like independent on which row it is on, it's pretty much you're assuming you're blank of sterile media. If it's non-sterile, like if it was contaminated, then we expect a difference in absorbance readings thereafter. But nonetheless, and that's post-incubation. Because this incubation, even though it goes up in time, you're primarily focusing on what you see at a specific table or what you see in this microplate reader for a single time point. So for each row, you have two reps, just like reps that we've done in the past of UV or at least of other, class exercises, right? So think about like even when we talked about osmotic pressure, pH, temp, we had controls and we had dupes, right? Like replicates of five tubes, five conditions, 10 tubes for the temps, right? So in this regard, for thermal death time, we actually had them for rows A and B at 50 degrees and C and D or C and E at 65 degrees. And they served as duplicates for the two sets of rows or like pairs. But the important point is that so from column 2 onwards, there was inoculations that repeated every 3 to 4 minutes. And I say every 3 to 4 because if you can consider the fact that this was 5 minutes for many of you guys. Actually, let me just go forward with the understanding that you guys can understand that, for each column, you have, imagine this. The tubes, the flasks are, either on the water baths or just taking them out. And then following the 0 minutes, you would have essentially used a pipette, place them into the wells for the two reps for each flask, be it for A and B for 50 degrees or C and D for 65 degrees. And then every 3 to 4 minutes, I would have continued to ideally do that. I say to 3 to 4 minutes because it's also pragmatically very difficult, or it is something you want to consider, or like just maybe bear like to like I understand it's like you would want to do it at every 3 minutes, right? To see the equal incremental change across time. Um, but you also want to see like and with that in mind, let's just assume, right? You would see thereafter a proportional change, ideally, in semi-log, right? Because remember generation time, you went with fixed increments in the microplate reader every 15 minutes, ideally for viable counts every 30 minutes, right? Till the end. And so the idea is that you would see when you make the points, when you plotted out those points, you would have made a line of best fit even though in that way it was, and indeed that was the case. In our case here for the TDT and D value analysis, your log plots, semi-log plots would be in the inverse, right? Because you expect as, just like how we talked about UV exposure or temp exposure, as you increase the exposure to extremities for bacteria to grow in, bearing in mind that you know the optimal conditions, if it is extreme, right, at a very high or very low pH or very high or very low temp, then you could expect over time that there is a reduction in growth and essentially that they remain in death phase or they're essentially passed. And so that's where you have the slope going down. That's where you would expect the points going down and the line of best fit showing the slope that goes down as well, or down in this direction. And so when it comes to understanding your D values, at least, right, TDT, I've mentioned before, it's the time it takes to essentially reach zero or no, like technically one bacteria, which for the log plot is going to look like a zero, or technically a log value of 1 because it's going to be 10 to the minus 0. So essentially, pretty much where it reaches roughly the x-axis, and maybe we'll look to confirm, like to see the description in the lab manual. But I believe that pretty much ends off the results because this is pretty much looking from what we described earlier from week 7. So let me just briefly go over then. Um, Yes, Alhamdulillah, Alameen, TDT, as shown here. So let's have a look just so we can all understand this better. To quantify the time, TDT, thermal death time, or the time to understand this quantitatively, this is the time that is required for a specific number of microorganisms uh, to be killed or to pass, sorry, um, at a specific temperature. So ignore the temperature point just for now. You look at this curve, right? At any temp, 50, 65, 40, 37 degrees, right? Exposure, though 37 probably would be a slope that's null or even going up. But let's say it was an extreme condition, pH, temp, salinity. You're going down. In this case, because we're focusing on thermal death time, it's focusing on a specific temperature. But the idea is the time that is required to essentially go down. So think about it for a moment, as shown here. And it's a bit detailed, but the thermal death time curve of a hypothetical culture with an initial cell density of 10 to the power of 6, right? You guys know why it's 10 to the power of 6? If you know, answer in the comments. Like think about what log refers to here, right? Or how that influences your population density. But you know this, let's say at 60 degrees. From the graph, it could be determined that the population survives up to 60 minutes, right? That's where essentially it reaches the x-axis, or sorry, the end of the y-axis. So the idea is that then the thermal death time would be assumed. Technically, you want to be more precise, in fact, right? It could be 59 minutes in the sense that, because it's very hard to tell here, where you actually have no more, like the time that it takes for a specific number of microorganisms to, die. If you start off with, let's say, and I want you guys to think about this. Let's say 100 bacteria, and it goes all the way down. In this case, let's say one. So this is actually 1 million bacteria, right? So 1 million cells per mil. It goes down in 60 minutes all the way to zero. That's technically 10 to the power of zero is technically one bacteria remaining, right? So that's why I want you guys to think critically. Essentially, the time that it takes to go and reach one bacteria is your thermal death time. You want to be more precise because when it reaches the x-axis, it's going to be in a log form at least a zero, you're assuming, unless you're being very particular and you can actually notice a small difference, right, between 4 to 2 being a little shorter than difference that's longer from 2 to 0 here. The idea is that eventually, once it reaches the x-axis, the difference is essentially your thermal death time, or the TDT value. And then for your D value, that's the time that it takes to go 90% down, which I think is shown better in one of the following slides. Oh, I think it's so essentially like maybe to make it easier here. Let's say this was 10 to the power of 5, right? Right here. So the time that it takes to go from 10 to the power of 6 to 10 to the power of 5, and you can interpolate from the curve, from the y coordinates all the way down, that period of time, right? And we're assuming that these are equal increments. So this would probably be about 10 minutes, right? So your D value is assumed to be 10 minutes, i.e. the time that it takes to go down tenfold or 90% in the population, because 10 to the power of 6 to 10 to the power of 5, that's going from a million, or like, I hope you guys understand, it's a tenfold decrease. Kind of, like think about like the serial dilutions going 10 times down, right? So 10 times down, you're essentially, that gives you your D value shown here. And then the other implication, just before we end off, is understanding them as you change the differences in, temperature, right? As I mentioned earlier, differences in temperature impact then the TDT and D values proportionally, i.e., if it's more extreme, right, your condition now, then you expect the TDT value to be shorter because the slope is going down more quicker, right? And so if it's going down quicker, then essentially it'll take shorter time for it to reach 1 or 0. And so essentially that's or one bacteria left, and that's, or at least to reach the x-axis, and that will be essentially your TDT. The contrast is with the D value, essentially it's a tenfold decrease, so that as well with a more steep slope, even if you go down tenfold on your y-axis, which hasn't changed, the difference now in your x-axis is much smaller, right? It's compressed more, and thus that depicts a lower d-value as well. So think about how those different components relate, and comparing and contrasting these results with what you've done from the generation time assignment. And so inshallah ta'ala, I hope that makes sense. And that actually moves us to week 9. If not, let me just compare. Um, the last component, maybe I'll touch upon for understanding your d-value. You can also use this formula, which is like the inverse that we've described from the past, in that think about it like this. If you go up here, right, your TDT is your time. And actually, I want you guys to understand this a bit more critically. And I say this because think about it like this once again, right? Your population is going down, right? From X, in this case, X to Y, Y2 to Y1. That's technically, what you're seeing here. Y2 is technically X, your initial population, right? Which could have started out at 0, right? Because 0 minutes, i.e., before exposure to heat. And then essentially Y is, 10 minutes, or actually not 10 minutes, the time that essentially the cell density that it was post heat, right? So you don't have to necessarily know the time range or mention the time range there on the right here, because it'll be mentioned in the top. So think of it like this. Let's say, right, if you, let's say, did this analysis of 10 to the power of 6, you literally put 10 to the power of 6 as x, or y2 in this case. And then if you actually think about like calculus, this would technically give you 10, this value itself will give you a value of 6. So log 10 to 10 to the power of 6 will give you a value of 6. And then let's say we did time minus 10, and then your y value, let's say, was the very bottom, making it simpler. So 10 to the power of 0, or 0 in this case. Then 10 to the power of 0 as like your base exponent would give you a value of 0, right? So that's 6 minus 0 is 6. And then the time, the duration, which we actually know is 60 minutes here. 60 divided by 6 is 10, right? Meaning that your d value neatly is 10 minutes. I'm saying this just so you guys understand this concept, right? It makes sense, right? Because as I just talked about, when you saw the previous slide, inshallah ta'ala, you can appreciate that it's going down tenfold every 10 minutes, right? Even if you saw it from the lab manual, it's the same diagram. You're going down every 10 minutes. So this is from 10 to the power of 6, going down 10 times less, 10 to the power of 5 at 10 minutes, then 10 to the power of 4 at 20 minutes, then 10 to the power of 3 at 30 minutes, then 10 to the power of 2 at 40 minutes, then 10 to the power of 1, right, at 50 minutes, then 10 to the power of 0, which is technically 1, at 60 minutes, right? So I just want you guys to understand that those increments are normally expected to be equal and to be representative or proportional across time. So even though that formula shows you that your starting value or cell density could be at 10 to the power of 6, right, just before you added in the heat, it technically could even be here, right? If you know the two separate points, just like how we talked about from the past, right, on your line of best fit. If you know two separate points that are indeed or on your line of best fit after you plotted out the results on semi-log graph paper, then inshallah ta'ala, you should be not only able to find what you would expect, let's say, from our previous assignment in generation time, but even in this case, you could find out the D value, right? And I think that will give you a good indicator of what you could expect therein, right? In terms of understanding how, or the relationship you would have for TDT, or like the relationships you have across temperature conditions for the growth of your bacteria across density. So bear those details in mind. And then, yeah, pretty much a very simple, calculation that you guys could consider. So think about these different components, and I believe that does conclude our analysis of week 8. So let's go to week 9, inshallah ta'ala. And I know we spent a bit of time week 8, that's okay. Inshallah ta'ala, we're going to go now to week 9, second last set of labs. And subhanAllah, it feels like it was already done so recently. But, like recall differences between selective and differential components, right? And think about actually what we've talked about, right? Do a little bit of a recap. Pause the video or like think about what I've talked about thus far. We've looked at many different types of differential stains or different stains, different techniques, different, apparatuses, methods, and different extensions, limitations, applications. And I hope you guys can appreciate them in this integration of our Golden Lab series. So you have catalase, beta-lactamase, those exist as biochemical tests, separate component to understand, but essentially you're now being able to identify specific properties of bacteria biochemically. That could be the enzymes that they have, that could be the different components that they have extracellularly. Remember the capsule stain, or intracellularly, the endospore stain, right? Or gram stain in terms of the thickness of their peptidoglycan layer and the implications that those all have with UV exposure or biofilm formation, right? So a lot of these details are connecting from our first set of review labs to our final set of review labs today. So I hope you guys have been able to actually integrate these concepts uniquely, diversely, and sincerely for his sake. So, and then with that in mind, inshallah ta'ala, we'll continue further. Right, we had 3 different types of plates, mannitol, salt, agar. I call it the MSA plate for short, or in fact, it's quite interesting indeed. And you have the different components. MSA, the MSA plate has 7.5% NaCl, right? So it's highly saline, right? It's highly concentrated with solute NaCl. You have mannitol, another type of metabolite, and you have phenol red, mannitol exists as a carbohydrate. And what you want to highlight here or appreciate for the understanding of the different components is, you know, before you even see the plate as considered selective or differential, what importance or what roles do these ingredients play, right? The salinity, the increase in salinity means that it restricts only certain bacteria to grow, i.e. is selecting, just like how we had for the BLA gene, resistance for ampicillin for transformation of HLB101 or HL101 E. coli, right? We're selecting here for only osmotolerant bacteria, right? I want you guys to think about that because that already gives you a hint, right? With the extremities that we've talked about from our previous week review, right? For week 8, week 7, or even week 6. So think about these concepts. And then fermentation, right? Mannitol, right? It's going to be metabolized, right? And when normally fermentation or when you have metabolism, bacterial metabolism, you have with, carbohydrates being essentially reduced or oxidized and essentially reduced, you have therein um, the reduction in pH, right? So with that, you have, hydrolytes increasing in solute concentration, interestingly. But what I want to highlight is that this phenol red, this dye, serves as a proxy, right? Because now think about it for a moment. Mannitol is just differentiating bacteria that either ferment this sugar or not, right? So if the like think about it like that, if a bacteria doesn't metabolize mannitol, that doesn't mean that it doesn't necessarily survive, right? It just doesn't metabolize it or ferment it, right? It could, like in these plates itself, it still has other, perhaps what you expect, and maybe just look at LB, agar, nutrient-rich medium. You expect there to be other nutrients that it can metabolize or grow on, right? So the idea is, or sorry, not grow on, but just to metabolize, right? Or metabolize and reproduce. And so I want you to think about it in this regard. Mannitol as a metabolite just serves to essentially indicate with phenol red whether or not, or like essentially if bacteria are able to ferment this metabolite. And what I mean is that, okay, so if it's able to, like, think about the bacteria that you see here. If it's able to grow, and see both are able to grow, right? Meaning that they've been selected from the NaCl. But notably, you have differences in color, right? So the mannitol is able to essentially, if bacteria are able to ferment it, pH goes down. The phenol red indicates a change in color when the pH goes down, right? So it goes from red at a pH that it serves at around 6.6, let's say, goes down to, let's say, 3 or 4 in pH. That color, that change in hydrogen concentration essentially exhibits a change in color, which goes from like pinkish red, shown here for Staphylococcus epidermidis, to yellow, right, for Staphylococcus aureus. Once again, it's highlighting the change in different species fermentation pathways or abilities to ferment certain metabolites or sugars, right? And so you could see and predict then aureus, S. aureus, is able to ferment mannitol, whereas S. epidermidis is not able to ferment mannitol, right? So I want you just to think then, or sorry, let me go back. Mannitol and phenol red, because both serve to couple the indication of bacteria that are able to ferment mannitol or the sugar. That just serves to have a differential, component, right? That means that it's differentiating. Either these bacteria fermented or not, right? And then, so that depicts a differential set of components. The mannitol with its diphenyl red. And then NaCl, once again, selects for bacteria that are able to grow or not grow. So I hope that gives you an idea to appreciate that then these two, all three components combined indicate both properties can be served in a single plate of selective and differential components, selective and differential biochemical properties, right? So they are able to be, let's say, these two bacteria are osmotolerant, but only one of them can ferment mannitol and the other cannot. So this hopefully gives you an appreciation that you can actually, in a single plate, differentially and selectively compare different traits biochemically. Like you could do this of not only like selectable markers of NaCl, you can add in ampicillin with the arabinose operon, right? You could even use a lac operon. You could use so many other, you could use discs with ampicillin. And like, I'm just saying this as an example, but there are so many other like creative, analogies or permutations that you could actually analyze other properties of bacteria using such similar or some unique selectable and differential properties or components of media. So that really, I hope, gives you a rundown of the setup of the lab, and also, inshallah, to highlight importantly in your methods that I know we dealt with 3 sections to keep one as a control, right? Avoiding cross-contamination as shown here. But bear in mind that when you did these cotton swab streaks, that you didn't, spread too close to other sections to run the risk of also cross-contamination once again, right? Like here. So even though that this is the control, just bear that in mind. And then the McConkey agar plate, I'm just going to highlight that you have your dye, right? Crystal violet. We saw this for gram staining, darkish purple, right? And you have the bile salts, right? And so bear in mind that, and it's shown here, crystal violet so actually, I want you guys to think about this. Crystal violet is a dye. Lactose is a metabolite, it's a sugar. Bile salts is a salt, right? So think about it like this. You have a dye differential, right? Differentiating, as we saw, gram-positive, gram-negative bacteria. Lactose is a sugar. And that also serves a differential purpose with neutral red. And then you have bile salts, which is selected, right? Selectable for only certain types of bacteria to grow, right? Interestingly here, it actually inhibits gram-positive bacteria, with crystal violet being taken up by gram-positive bacteria, right? But it prevents the further division of the cell wall, right? And bile salts are also generally more toxic for many microbes. So bear these details in mind. They'll be highlighted also in the manual, but essentially coupled with biosalts and crystal violet together. Um, sorry, let me just confirm. So the biosalts essentially with with crystal violet in this case serve to select for the growth of gram-negative bacteria or selects for only specific bacteria to grow. Bacteria that are, by the way, osmotolerant or at least, and actually, we'll have a look. I don't want to pressure you guys, but essentially, you have those sets of bacteria that are able to grow, and lactose serves as a differential component, i.e., with a reduction in pH, just like before, neutral red could go to a more yellowish hue, especially in the smear itself. So that would give you an indicator, right? Because the pH it can actually go down post incubation and so if it ferments, right, if the bacteria ferments lactose, then it can actually reduce the pH of the media or the section, and that leads to having a reddish or like a more interestingly, a faint red or even to a colorless um, a color like a low hued stain or like a dye, like a hue, to essentially having a more pink red hue that is depicted here for quadrant 4. So I want you guys to think about these details, and then you can contrast, and actually you could perhaps predict, right? Like what types of bacteria perhaps grow here. What did you expect for E. coli versus Staphylococcus epidermidis, right? And so once again, the neutral red with the lactose differentiate bacteria that are able to ferment lactose, and the crystal violet coupled with the bile salts are able to select for the growth of only specific bacteria that are gram-negative. If you, let's say, did not have gram crystal violet, then it's a bit different in the sense that you can have technically the growth of gram-positive bacteria, and that's actually what I expected. So we'll have a look to confirm, inshallah ta'ala. But nonetheless, have a look here. Gelatinase, we talked about this. If the tube has, actually, before I go there, um, let me go to DNase. And it's another enzyme that's actually very important to understand because it connected us back to the conjugation and I believe transformation. Um, I want you guys to think how and why. Think about DNase, right? Or think about what I've talked about actually before then. Right? So it's testing in this regard for the presence of DNase, right? And your bacteria. And so the dye includes, or the media includes, sorry, DNA and methyl green. And so methyl green is a cationic. Think about like the oxychrome, right? We talked about that before, but in this case, it's positively charged, which would bind to negatively charged DNA, right? And give that bluish hue essentially. But what's important is that if there's a breakdown of DNA around the periphery of your bacteria, i.e., if it has DNase, then you expect to see a halo and the clearing of that bluish hue because that clears or depicts the clearing of DNA. So the clearing of DNA, clearing of the blue hue, indicating halo, indicating DNase being present in the bacteria itself which is able to, and that's something to assume. It's an exozyme, meaning that essentially it could come out, it could essentially uh, cleave and hydrolyze DNA outside of the bacteria. And so that's something that you want to keep in mind. And so, inshallah, that makes sense. I'm going to go back to Gelatinase, or if you look at Gelatinase, yes. Sorry, give me just one moment. The tube itself has the media that includes gelatin, right? It's a protein found in connective tissue. And something that to point out is that it's solid, as you saw from the very beginning of week 9 material. it was solid at the beginning, right? You put a smear like the motility test, and post incubation, you could see changes in viscosity, right? If, sorry, let me go back. If it has the bacteria, let's say after you smeared, inoculated the tube half inch or halfway in, if it has gelatinase, which is also another exozyme, or another enzyme, rather, if it has this enzyme, then it will be able to essentially break down, right, gelatin, and meaning that the porous nature increases. And like essentially, because if you think of connective tissue like a matrix, now the connections, right, of the tissue itself, or essentially the the solubility or the integrity of the media itself decreases, to which the viscosity increases to be what you have here, right? So it becomes liquefied essentially, in the case that gelatinase is present. So that's something you want to bear in mind. Bear in mind that you have B. subtilis versus E. coli, and you had a control tube. So if let's say your control tube showed liquefaction, I want you to think, what do you expect? What could have happened? What could have been done wrong, right? So think about that. Consider these components or responses to these questions at least as well, and the implications that these enzymes, biochemically have on your other analyses from bacterial, microbiochemistry, essentially, an interesting subfield that we essentially were, or something that we're just briefly touching upon, right? We looked at catalase, cephinase, or beta-lactamase, gelatinase, DNase, right? So four enzymes thus far, right? And I want you to appreciate even the other ones that we've touched upon, photolyase, hydrolase, in another way. All right, so think about like the different components, even outside, or following these slides, or even following the pages in the lab manual, or even your lecture slides. And then biofilms I talked about. Let me just see in terms of timing. There are three main other components I just want to highlight. Um, Biofilms, briefly, changes in biofilm appearance or biofilm generation or formation is exhibited by a change in the staining of your tube, right? It has, remember, these tubes, like in the case of Staphylococcus epidermidis, it's a gram-positive bacteria, is able to, imagine grow, right? Think about the growth patterns, and thus, if there's a lot, an abundant amount of bacteria, right? Without UV exposure, let's say, or like if as a control in this tube, then you can appreciate, after you've rinsed, after you've resuspended the bacteria, or even if you, like let the the slant maintain the biofilm on the surface or on the inner surface of your tube, then you can appreciate from the culture after you've decanted, rinsed, and with PBS, then with crystal violet for 10 minutes, and then rinsed with water and dried, you would have appreciated that you could see between your control and your Staphylococcus epidermidis slant, changes in appearance, right? And the residue that is left. That residue, as you can see from the manual, has important components that I hope Bit and I will talk over. And then you have the Snyder test, a virtual, but something I touched upon indeed, and I want you guys to think about as well, indeed, for your effective practice. If you haven't sent it, that you could see a clear change in different in color, right? From forest green in this case to lemon yellow. And you could see three highlighted components: dextrose, glucose, metabolites. So think, is it selective or differential? Bromocresol green, think about it, it's a dye. Is it differential or the selective component? And then pH, right? It's at the extremities. So think about it as well. And even consider 25 degrees as well. That's also an extremity per se, if depending on what bacteria you're using, right? So think about what the bacteria are of interest. Consider its optimal ranges, which you can see from the manual, and you can hopefully appreciate, if not if you explore online, the different components and the applications that they have. So that's what I want you to understand. Usually it's used to essentially determine the susceptibility of dental caries or cavities based on the abundance of lactobacilli or other bacteria, right, that are able to ferment components of one's teeth. And that also connects us to the biofilms because the presence of these bacteria on surfaces, just like how we talked about antiseptics, that if you had those bacteria that actually grew without the presence of, germicides, then they could coat a surface like a glycocalyx on themselves, but also across the extracellular matrix that they could have, right? So that connects us even back to the connective tissue component of gelatinase. But just imagine hypothetically you can have an entire surface of microbioflora, right? Which connects us to our own bodies. But think about like this as a lining of bacteria secreted products. That could be of sugars, like sugars on their surfaces, that just were like removed from the surface of the bacteria, but are now on the residual surface of your tubes internally. But you can see like the degradations, right? If you, let's say you wash too rigorously, let's say you pelleted and a lot of the bacteria was seeped through, including the pellet, or let's say another idea extension in this case where you have like these intermediate colors, as I just hinted before. What if you had like UV exposure or high temps, right? And that in turn could also influence like, the maintenance of your biofilm. So think about like the biofilm as a marker for the quality of the communal proliferation of your bacteria, which is so interesting to connect because we've been looking at individual bacteria, right? Viable counts, counting specific colonies that depict colonial forming units. Um, to like Alameen looking at the unknown assignment, different types of bacteria. Now, I really hope you guys have been able to appreciate, the diversity of approaches. But now, with lastly the blood agar, I want you guys to see the different components: beta hemolysis, alpha hemolysis, gamma hemolysis. This is a selectable, or sorry, differential trait in which you're able to see changes, right, in the bacterial growth. Bear in mind the media itself, right? That if, let's say, it was hypotonic, that it could also exhibit a selective nature. But essentially, you have sheep's blood, hemolysis substrate. Have a look at these videos because they also give some hints. But nonetheless, hemolysins can have grave effects, right? And different bacteria that exist as another set of enzymes that could break down, depending on if it's a beta, alpha, or gamma, the changes in color depict the the component or the subset of hemolysins that they have of your bacteria that can thereby impact RBC growth or survival. And so that's where beta hemolysis can be quite detrimental. And then the impacts of gamma could have less so, but nonetheless something that you want to consider for understanding the selectable and differential natures of different bacteria. And then the conditions that we've already described, the different growth patterns across temps, pH for different species and classifications, even at the extremities, from thermal to hypothermal files, from sacral to trophs to sacrophiles. So many different types of components. And I already talked about osmotic pressure. And just to appreciate, you have non-halophiles as non-osmotolerant. We have also a diversity of like halotolerant to halophiles, more like the halophiles to extreme halophiles that depict osmotolerance. And then I described as well the D value that can be shown here, even outside of the initial cell density post exposure of heat. So that way you could see essentially a tenfold decrease that depicts the D value. Now I'm just gonna go back. I do wanna, should I not? I don't know if I have time, but inshallah ta'ala, I just briefly wanna maybe, okay, let me see. We have our last lecture to go through, week 10, which is the lysozyme assay, and pretty much incorporating everything that we've looked at thus far. So let's go briefly now to week 9, just to with the lab manual, so that way at the end we can just compare what we've garnered, and inshallah ta'ala conclude. Briefly though, so we have once again selectable versus differential traits, right? And I hope you now are able to understand the classifications of all and hopefully see the applications, the fruits of these different, or like the integrative nature and the biochemical appreciation or analysis and extensions of this project for you guys, or this exercise in the lab itself. Yes, so mannitol is a carbohydrate. We mentioned sodium chloride, selective component that we talked about before. Chemical germicides, control, interestingly, um, control being here, sorry. And then B being Staphylococcus epidermidis, Staphylococcus aureus. And this is, by the way, the type of detail that you guys want to know, being mindful of the different components. The Snyder, I know this is a nice one, but nonetheless, think about the application. It could be something useful to consider for you guys to understand, like for the lab exam and beyond. Like if you're interested in, let's say, dentistry or other lab biochem courses, right? You can take a bit of your saliva, put into a liquefied Snyder test. You can notice how it's at 50 degrees, so that's quite a high temp, high water bath. And then after 24 to 72 hours, you can actually see if there's a prominence of lactobacilli yellow coloration, right? So that's something interesting. Then you have the ability to ferment lactose from a conky agar plate. And this is found, and bear in mind the different conditions. You have Enterobacteria CA, which is one of the members includes E. coli. This Escherichia coli is one of its members. And the idea is that you have, the different components that we mentioned, red, neutral red, lactose, bile salts, crystal violet. So I want you guys to understand, right? The crystal violet, interestingly, I understood it as to be differential because, like, obviously we've used it in the past to differentiate, gram-positive versus gram-negative bacteria. But just to clarify this just a bit, bile salts and crystal violet together serve a inhibitory role, and it's just something that I want you guys to maybe explore a bit more to understand, crystallize this as well to with understanding. But essentially, the bile salts and the crystal violet will couple together and essentially inhibit and think about it like this: when we smeared the gram staining, right? We'd heat fix and then do crystal violet staining. But these are now dealt with in live cells. So crystal violet as a stain, as a dye, coupled with bile salts, you can imagine that it's going to be selectable in a sense that, if crystal violet passes through, hypothetically, now with NaCl, right? Think about the osmotolerance, tolerance, right? Think about like gram-positive, yes, or gram-positive bacteria, but think about it like this. If those salts are now within your bacteria, to give you guys a better understanding of this, think about osmolarity, right? Now that the salts are actually able to enter within your bacteria, think about the impact. The water is going to gush towards inside, internally into your bacteria and lead to lysis, right? Because it's a thicker peptidoglycan layer, or it's going to be more obstructed. And so the essentially um, integrity of your bacteria is now in jeopardy for gram-positive ones. So that's where gram-negative, existence exists, or like that's where the selectable, or that's where gram-negative bacteria persist. And then the difference, just to clarify, from the gram-negative bacteria, those that ferment lactose, interestingly, depict a reddish pink hue, which can be shown here, and then a non-lactose fermenting bacteria, which you can imagine from what we've described as a hint, is essentially will give you a less than reddish hue. So it would be like Staphylococcus epidermidis, less of a hue in range. If you have seen though a reddish hue for be it E. coli or stuff, well, actually, let me clarify. Would you see Staphylococcus epidermidis? Yes or no? The answer is no, right? So let me just to highlight this, that if you, let's say, from the Enterobacteriaceae genus, if you had other bacteria, let's say those that were gram negative, but let's say essentially did not ferment lactose, then you would probably see a clearish hue that appears more yellowish. So it doesn't necessarily mean that they do ferment, but rather they don't. And the neutral red actually exhibits that change in color from colorless to red. Think about the motility test, or just in this case for fermentation of lactose, a pinkish red hue. And then gelatinase, we spoke about. Interestingly, there are other bacteria that you may want to know, like anthrax, for those that are involved in anthrax. Yeah, DNA hydrolysis. Um, we have other sets of bacteria that we're using, right? E. coli, B. subtilis. But hopefully you can actually appreciate that some sets of bacteria, even the Staphylococci family or the group, in the subgroup, you can appreciate that Staphylococcus aureus can actually, break down DNA as in DNase. Um, and likewise Serratia marcescens, but not other staphylococci. So it's a good way to actually appreciate different components. And then you have exotoxins. So we have the exoenzyme of gelatinase, or sorry, DNase, to now blood agar, exotoxins, hemolysins, leukocidins, right? That break down red and white blood cells respectively, and coagulases and fibrillysins, which, something by the way is connecting you to probably C23 and B12. So you can definitely see the applicatory nature of this lab. And then you have, the differences in clearing. For beta hemolysis, complete clearing exhibits lysis, complete lysis of RBCs, destruction of the hemoglobin. And I say hemo because think about like hemoglobin being a part of, right? RBCs. So, and, like when we, when oxygen travels throughout the circulatory system, right? And you can think about that as well, CO2. But nonetheless, like oxygen passes through, connects with, reacts with hemoglobin, or like connects with hemoglobin in the, sorry, essentially hemoglobin serves as this role in transporting oxygen and other gases using within RBCs. And so essentially the destruction of RBCs exhibit a clearing of the media and you see a greenish yellow hue. But clearly what you can actually appreciate is that for other components, and actually I want to know, yes, this is where it's mentioned. So the tryptic soy agar and the sheep's blood, essentially sheep's blood as with the RBCs, the tryptic soy agar helps to also exhibit that change in clearing. So that way you know if it's a full clearing, that's beta hemolysis, and that's a full lysis, or sorry, with the alpha lysis, it's essentially um, with these exotoxins that may lead to a partial disruption of the RBC membrane and the release of hemoglobin resulting in a greenish coloration. That's more like faintish uh, red or yellow. So it's more like a greenish hue. It's not a full clearing, right, around the bacterial colonies, which is the case also for beta hemolysis that you have our bacteria growing live or there. Then you have the gamma hemolysis, which is non-hemolytic, but essentially the medium appears non-altered. So you'll just see the streak. So based on what you see from your streak, if you see a full clearing, that's beta hemolysis, like a pure yellow hue, and then if it's a greenish hue, that's more faint, then that depicts alpha hemolysis, and gamma, where there appears not a really good streak. And then you have the different components of streptolysins that exist for streptococci. So bear in mind that that's for separate, if unless you're looking at these bacteria, they have a unique reference as being known as streptolysins, unique specification. In fact, of even more specifics, There's type O and type S. Type O refers to oxygen liable, and they mainly grow in anaerobic conditions, these sets of bacteria. And then streptolysin S is expressed in optimally expressed in anaerobic conditions as well, but is oxygen stable. So it could also grow, like if, let's say, streptococci has streptolysin S, even when oxygen is present, they could still optimally grow, or they could still grow the streptolysin, which can actually, as you can imagine, have impacts, right? For beta or alpha, whichever of the two these are that your bacteria is involved in. So bear in mind the differences. Beta, you could see the clear yellowish hue. Actually, the yellowish hue depicts the tryptus soy agar. So just bear in mind that that's essentially the clearing, the full clearing that makes the contrast with now the tryptic soy. Um, that's why you see it appear yellow. With the beta, sorry, the beta hemolysis showing a clearing, and then alpha with a greenish hue, which you can understand from the hemoglobin being released from with alpha hemolysis, and then the gamma as a non-clearing, essentially, um, non-hemolysis. So that's pretty much it, in fact, inshallah ta'ala. And I'm gonna go straight, forgive me for the delay, folks. Let's go straight now to our last set of exercises with the lysozyme assay. Lysozyme, so we're now like in the physiological realm, right? We went to biochem in a bit, and we also went to physiology for some components like environmental conditions. But let's now look into the lysozyme assay. That's one set of final enzymes. And so you find in different secretory glands, regions within one's body, and other organisms, and tears, let's say of crocodiles, to saliva of a panting dog, or like ourselves when we sweat, even in the Suleiman Island frogs, right? So connects me with biology too, which is why I'm mentioning some, different organisms. But nonetheless, a lysozyme is able to break down chemical bonds in our cell wall of bacteria. So that gives you a hint of the type of bacteria as well, or the density and propensity of effect, the impact or severity that lysozyme could have on different bacteria as a substrate. So your bacteria serves as a substrate, gram positive, gram negative, catalase positive, negative. Think about the different traits or classifications of the bacteria across environmental conditions. And then your enzyme serves as a lysozyme with your buffer. And so you have different components that exist, with your blank having the buffer, and then the other sets of tubes, which will normally be done across increasing content, across your bacteria with increasing concentrations of your lysozyme. And then you would have plotted these values following the spec 20 reads, right? So you would use the spec 20, you would have blanked it before, and then so think about, the effects, right? Recall that you're breaking down bacteria. So if you're increasing the concentration of the lysozyme that breaks down bacteria, then you expect the turbidity to go down, right? Or even viable counts if you were to take them after, a set of, serial dilutions, perhaps. Or maybe not even say serial dilutions, but you would essentially assume a decrease or a lysis of bacteria, and thus decrease in turbidity over increasing concentrations of your lysozyme. And as I mentioned from our last lab in person together, what you can appreciate is that you can plot these values, and this is in Cartesian, i.e. regular increments, not semi-log, but with turbidity, what you can appreciate is that you're plotting them down like a curve, a smooth curve, and then you would interpolate, right? Based on what I've shown here. And I want you guys to do this. If you haven't sent it to me, you can include it as your comments, right? This exists as patient 1, patient 2, and essentially serial dilution 1 and serial dilution 2, right? So think about it like this, and these other values depict your absorbance readings. So you would go here, Look at the absorbance readings. Let's say 0.43. Go down from y coordinate, go down all the way to the right to the curve. And we reach the curve, we go down to the x-axis. The x value looks like maybe 750 nanograms per mil. Bear in mind units, bear in mind your axis, standard curve title, figure caption right underneath. And that, subhanAllah, that the details are quite hopefully explanatory. That you now have that 750 nanograms per mil, and that's for your diluted, 10 to the minus 1, sample, but then you multiply, right, by 10 to essentially give you the undiluted concentration of lysozyme. So that would be essentially 7.5 micrograms per mil, right? So think about like that, you'd be able to essentially appreciate, how many, you're not using a mass balance, but you're actually depicting approximately under some limitations or assumptions that, those are exhibiting viable cells, right? Because the bacteria, they're essentially dying. And so you're actually looking at the turbidity as a result of how many cells are thereby living to give you a proportional read for absorbance and a proportional effect of the lysozyme. Because there could be other modes in which, let's say, the bacteria are able to resist, perhaps, right? And so you want to consider, or maybe just consider from what you've seen, the different assumptions that you would take into account, right? Turbidity, is it indirect or a direct method, right? Think about what I've mentioned, and hopefully that will give you a better understanding. And so you would repeat the same process once you've made your curve, right? Make it smooth in this case, nonlinear. Remember that if it's like generation time semi-log or TDT semi-log, it's a line, best fit, and then you would plot accordingly. And so just bear in mind that after that, you interpolate, you go down, reach the x-axis, find out the concentration, account for the dilution factor divided by the negative exponent. So when you divide by a negative exponent, then you get the true value that's higher, either 10 times more here or 100 times more than the concentration that you find from the interpolated points on the x-axis. And so essentially that's what you would get. And for patient 1 and then patient 2, you would essentially get the mean for both because technically it's like viable counts, right? In the sense that you have two sets of dilutions, and then you would correspondingly depict, let's say this was at let's say this was, 3 micrograms per milliliter, and this was, let's say, 4 micrograms per milliliter. Then the average for patient 1 would be, let's say, 3.5, right? Micrograms per mil. Whereas this, let's say, could be, hypothetically 6 micrograms per mil, and this could be, 7. So then micrograms per mil, so the average would be 6.5. And so you can actually depict then, okay, the relative abundance, like patient 2 maybe has more, patient 1. And then, comparing between time points, time 1 versus time 3, greater incubation with lysozyme, what do you expect? And that pretty much concludes, because I combined second, third, and fourth slide. That concludes pretty much what we have. We can go back. I want you guys, if you haven't, by the way, once again, please either send this to me. I recommend you sending this to me as well, of course, for your post lab for week 10. Or actually, what you could do is go through the comments, write down, your comments on underneath the YouTube video, and share what you liked, what you learned, or what you learned new, or what we clarified for student concepts. And then inshallah ta'ala, I want you to also highlight, any outstanding questions that maybe you have as a second point. And then the third component, any outstanding questions that you may have missed to answer, right? And as a fourth point, if you can, this is supplemental, because each seminar you want to have 3 questions or 3, modes of discussion. The fourth point is asking questions to other students, right? Maybe you can actually, serve it as a discussion board. Ask questions. Like let's say, you found another set of lysozymes, lysozyme B, right? And it essentially has a, it has a reduced effect, in bacterial lysis for, let's say, gram-positive bacteria, then what do you expect would happen to this curve, right? And proportionally, what would you expect then of your absorbance readings, right? So think about, think critically, think about like these types of questions. I really want you guys to um, expand your breadth and knowledge of the concepts. I didn't touch upon the biofilms, clinical biofilms, but we did actually for week 9 material, let's go over it. Essentially, you can have like these matrices of hydrated extracellular polymeric substances like polysaccharides, proteins, nucleic acids, and lipids, which bacteria can feed on, right? And so you can actually have them build off, right? Lactobacilli that connects us, or like with the Snyder test, or with our other, even our plates, right? Because the plates, the bacteria are growing. They're able to essentially, with the media, essentially absorb, take up nutrients, and release exozymes, release like the DNase, or release certain, waste products. And so essentially, they can technically, if you accumulated those plates, over multiple weeks, not just like for a specific time point or even 24 hours overnight, but let's say 3 to 100 hours overnight, then you can imagine a huge mass of cells. Similarly for a single tube, right? So that's something I want you guys to think about. The extensions, right? The use of crystal violet, right? In this case, is the differential. Answer is yes. And then so think about like the other components that I mentioned thus far, and we're almost done concluding the lab. So I'll spend about 6 more minutes, or however long, how many more minutes Allah subhanahu wa ta'ala wills, however much Allah wills, and is most pleased with the night time. Lysozyme for week 10 is also known as a neuraminidase, and this is actually something to bear in mind because this refers to those that are interested in research, the essentially glycolysis of a specific glycan, which is like a proteoglycan found on the surface of your bacteria. So essentially, I just want to make it easier to understand, but these types of lysozymes exist within ourselves, internally with our saliva, externally with the tears, the mucus, human milk, breast milk. They're very beneficial, right? Reducing microbial infection by the Almighty as well, or by His will. So this is something that you want to, appreciate and extend to understand the implications of. I mentioned this as well for our last practical, right? These are the 1,4 beta linkages, or I didn't mention, I asked that as a question, for between N-acetylmuramic acid and N-acetyl D-glucosamine or glucosamine residues in peptidoglycans, which is a major component of the gram-positive cell wall, right? So we're connecting back full circle to week 1. SubhanAllah. So I really hope that you guys have actually come to appreciate the different connections that we've been making and right the beneficial nature of this lysozyme, which can be beneficial to understanding its concentration in ourselves to see how we could better optimize or better increase its production so that way we could do, and this is just like very hypothetical, but something that you can appreciate that if understand the techniques and understand the applications of certain components of bacteria or of enzymes, then you could see how medicine could be quite applicable or replicable to your understanding and appreciation of microbiology and beyond, or even of the sciences and beyond. I'm sorry. Or to, explore to be more critical and analyze beyond the realms within and beyond the sciences indeed. Yeah, so actually, believe it or not, this was the prep of the saliva samples. You have the appendix, aseptic techniques, sterile methods, right? So this actually connects us to the reflective practice. What new concepts, connections were you learning, right? In the last question, right? I want you guys to look back even through the appendix if you can, right? Focus on what I've touched upon, the material, right? And I want you to once again, like Remind yourself that, just like for an FLR, you have an intro, you have your flow charts, in that case, the methods, right? Um, from your lab notebook. Um, you have your results, you have your with the figure captions, the tables, the calculations, observations, and then the discussion includes your expectations, literature results, like your connections with your interpretations of your observations, the limitations of your observations. I mentioned expectations or observations, your conclusions, the extensions. And by the way, I didn't like understand the impact of your limitations as well. Very important, right? If you don't follow aseptic technique, how well can you perform the many other like any sensitive, experiments, right? Or like other techniques that we've learned thus far. Or, if let's say you didn't add in the right volumes or you didn't resuspend your bacteria, right? These are things that I want you to think about and the connections in which how they can imply certain results in your lab exam and for maybe even just before this in your observations from our previous exercises and labs slash practicals with each other, right? So think about those details, and with that, describe your conclusions, understand what you expect. What are the implications of what you expect? What are the applications they're in when you have specific questions that are case scenario based, right? Or the applications of understanding how microbiology can be used, can connect beyond even biology or general biology into biochem, into nanophysiology, nanochemistry, to human physiology, to biochemistry, to neuroscience, right? To so many fields that perhaps maybe you haven't explored as of yet, but you come to appreciate, when you're interested, like as you explore medicine, research, industry, and any other, explorations that you're interested in. Bear in mind that you know how to use, by the way, micropipette. You see the highlights here in the appendices, right? This is 500 microliters from a P1000. This is 20 microliters from a P200. And then this is 2.5. I'm actually reading the top, so I wasn't looking here, but 2.5 microliters from a P20. And then I want you guys to, maybe create your own, right? Think about it like that. And, this could be a simple example for you guys to think about. I know that this is 200 microliters. I know that this is 1,000 microliters. I know that's 15 microliters, right? So, alhamdulillah, that's correct. And then 1,000 microliters is 1 mil. And just remember that, when you tip in, like when you press in into your micropipette, sorry. Actually, I hope that makes sense. Oh, well, but nonetheless, yes, like bear in mind that press the first stop to essentially go down into your pipette before you essentially ingest, essentially the pipette takes up um, your fluid, right? Your liquid of interest, or your chemical or your bacteria. So essentially, you go to the first stop of your plunger going downwards. And then when you go up, right, be it to inoculate, always remember aseptic technique with your tips, closing the tip box, with your lids, flaming with your tubes, flaming the lids, even for your beakers, like with the flasks that we've done for generation time, for example, or even with the necks of the glass pipette canisters, right? I want you like the bore pipettes. I want you guys to consider these and even the plates, right? Keeping them close by. They're not going to melt beside the Bunsen burner, but you want to bear in mind that you're not like letting it be an open exposure, just like with a UV analysis, right? So I want you guys to think about that. And then when you go up into another like tube or plate, and you inject full all the way down to the second stop until you reach the bottom of your plunger as shown here. So it's like the first stop here when you're about to take, before you take up, because you actually want to expel any air, essentially oxygen, that may be found in your tip. So when you're expelling the volume that's reached, let's say here at 50 microliters for a P200, then essentially you go down, and then you would let go to actually intake It's like kind of like inhaling and exhaling, air, right? In this case, you pressed downwards to exhale, and then you lift it up to inhale, like the media or the liquid of interest. And then when you're going down, it's like a full exhalation, right? Like a strong exhalation or like going down to the second stop. So that way you can make sure that you remove all of the liquid that you need to. And then the describing morphology, this is also something to understand, right? The inhibitory, I didn't talk about this, but bacteriostatic versus bactericidal to make it easier to understand. In your zone of inhibition, just understand this point for antimicrobial susceptibility test. If you have bacteria that are in your, sorry, if you have made your like post incubation, you have your discs and you have your zone of inhibition. In your zone of inhibition, if you essentially use cotton swab and then smear aseptically into a separate plate, subculturing into another like in a separate plate for further growth, then post incubation, if you find in that plate that there is indeed growth, then that exhibits bacteriostatic inhibition of your antibiotic on the bacteria. Whereas bactericidal means that there would not be growth. So I know that may sound different to understand, but understand that you have, like, if you have, so the problem is that around your zone of inhibition, you have a disc. And around that zone of inhibition, you may not know if there is bacteria so that are growing. So if they do grow, then that's bacteriostatic. If they don't grow, then it's bactericidal in a separate plate because that separate plate it should be able to grow in with new nutrient broth, fresh nutrient broth, right? That's rich, so they should be able to grow with the Luria bromoth agar. So as you can see, there's different types of agar as well, how they can be prepared. Very important understanding as well that the right conditions are met, aseptic technique even for the broths themselves, right? So that way you don't run the risk of any contaminants, even bacteria. So this is the Luria Britannia LB broth that we are often using, and you can make note of the different other medias or agars that we've been using for. And so, yeah, so you'd omit the agar in the case of just the broth itself, but with the agar, like the LB agar plates, then that's where you essentially have the agar included. You can imagine there's a lot of work that gets into, even understanding the different treatments, the tests to have resounding results, right? So these are things that you guys want to understand and hopefully appreciate within and beyond BIOC 17. Are there any questions? Any thoughts, components, criticisms, considerations, compliments? I really hope, brothers and sisters, that you've come to really appreciate the extension of the interpretations of our results all the way from week 6, in fact, all the way to week 1 to 10, bid'Illahi ta'ala. I really hope that you guys can appreciate the integration of these different components beyond what you can really perhaps bear to fully appreciate right now in the lab, right? Because in the lab a lot of things are high tense, right? I understand a lot of things are maybe appearing to be quite discreet, and my goal is that at no time do you guys feel like stressed out, right? Because the idea is that I want to make sure you guys feel comfortable in this space in which that many people may find detrimental in the sense that, or not detrimental, they may find it puzzling to be the only Black or Muslim or BIPOC individual, right? I want, or like for me at least, being a double minority, Black and Muslim, Muslim first indeed and only, that it makes me feel that I need to see more people for the sake of Allah subhanahu wa ta'ala, for His pleasure, to be in like such a space where they feel welcome, So I want you guys to feel welcome. I want you guys to feel content or appreciative of the the integrative potential of what you've learned from the past 10 weeks and technically our 11th year today. So I appreciate what you've been able to study, right? Be it from the slides that we've gone through from the manual, over 140-ish pages, right? Like there are many different techniques that we've described, and I really hope that you guys can appreciate them beyond C17. So with this, سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك والعصر أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. Jazakumullahu Khairan Katheera. Really appreciate it. And just want to highlight once again for your homework in terms of performance or like participation, answer questions, add them into the comments, ask questions to myself for clarification, or ask questions to your peers, which is even better, to pose, right, to get an understanding, to appreciate the content material, answer questions that are discussion based. And by the way, look back into the past from the very beginning, the differential stains, the gram stain, endospore stain, capsule stain, different classifications of bacteria environmentally with UV exposure, with the different germicides, right? The different impacts, the different modes of measurements, right? Turbidity, thermal death time, viable counts. So turbidity, direct count, and you know what I mean, right? Like the different experiments as well, TDT, D values, the Lysozyme Assay. There's so many things in the street planes, spread plating, mixed culture, pure culture, the different enzymes, catalase, gelatinase, antimicrobial susceptibility tests, the modes of antibiotics. I hope you can all like, even if it's just three different points from each of you, that would be really appreciated, inshallah ta'ala. And I hope that way you guys can do your best in this life before the next. May Allah subhanahu wa ta'ala make it easy in all of our tests. For indeed, in His remembrance do our hearts find rest. سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك. والعصر أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. جزاكم الله خير كثيرا. السلام عليكم.